Holler! Welcome back. We are talking about chapter 23, the continuation of chapter 23, evolution of populations and the Hardy-Weinberg equilibrium. Again, not to be confused with the sexual assaulter, Harvey Weinstein. Okay, let me share my screen. Share my screen. Oh, come on, screen sharing. There we go. All right, so we left off last time. Hold on, I gotta fix my screen. I'm on fire today. All right, so last time we talked about P and Q, and I just wanna do a quick reminder. So P represents the dominant allele frequency, or how often we see the homozygous, the dominant allele. For Q, we are looking at the recessive allele frequency, or how often you see the recessive allele. Remember, this takes into account that within a population, you have a mix of three possibilities: homozygous dominant, heterozygous, and homozygous recessive. And each of those kind of gets split up into a gene pool, which looks at every single allele in a population. And that's how we figure out allele frequencies. And microevolution is defined as a change in an allele frequency within a population over generations. And there are 5 main causes of microevolution that we're going to talk about now. We have mutation, non-random mating, migration, natural selection, and genetic drift. Yes, you have to all know all of these. Remember that we also talked about previously the 7 pieces of evidence for evolution. We have biodiversity, embryology, fossil record, vestigial structures, homologous structures, analogous structures, and molecular biology. You also have to know these 5 causes of microevolution, all right? And I don't have an awesome beefy ham situation for you guys, but what I do have is MNMMG. MNMMG. NMNMG. If you guys come up with a great acronym for this, please let me know. So these are going to be the causes of allele frequency changes in a population. Oops, let me change my. All right, so the first one is mutation. So mutation is a change in the DNA that can lead to a new version of a gene or a new allele, all right? And usually we are talking about, well, always, we are talking about random mutation. And that is often a result of, for example, normal DNA replication can have an error, and you might get a random mutation in that regard. You might have an environmental factor that causes a random mutation, all right? But again, if that random mutation is going to be heritable, it must be in the gametes. All right, so here we have a little picture of a population of little beetles, and they are all homozygous recessive. So we have an allele frequency, or Q, of 1, meaning that our P, or our dominant allele, is 0. Remember, those always have to add up to 1. When this population makes their gametes, their egg and sperm, there is a random mutation here, where one of the little A's in the eggs here turned into a big A. There was a random mutation that caused an allele change, right? Here we went from little a to big A. That created a new phenotype and a new genotype. Phenotype, we now have a little black beetle, and the new genotype is now we have a heterozygous individual. So if we calculate the allele frequencies, we would have our big P, or our dominant allele, or P, 0.05, and our little a, or recessive allele, which is Q, which is 0.95. And remember how we figure those out. We're going to take number of allele over total alleles, right? So here, you can make sure that that math works out, but it does. And so mutations can lead to allele frequency changes over time. That is one thing that can drive microevolution. The second thing that can drive microevolution is called natural selection, which you guys are already familiar with. This is where individuals that are better adapted to survive and reproduce, have babies, at higher rates. They do that because of those traits. And what we see is that the frequency of these helpful alleles increases in a population over time. So here we have a population of beetles again, and there is a predator, a bird, that is trying to eat the beetles. These black beetles are well camouflaged, so they don't get eaten as well or as much. The white beetles, on the other hand, are going to get eaten by the bird, right? So the black beetles are more adapted to their environment, and they're going to survive and pass on their genes. When we calculate allele frequencies, we see the following. Dominant allele frequency is 0.3. Recessive allele frequency is 0.7. After generations, we see that there is a change in the allele frequency. Now we notice that the dominant allele P has gone up in frequency and the recessive allele Q has gone down in frequency. This is due to natural selection. So natural selection can cause allele frequencies to change over time, and that can drive microevolution. For natural selection, I just want to point out that it can happen in three main ways. All right. So natural selection can shift phenotype distributions in three main ways. So if we look at a population of mice that have we're looking at fur coat color, and they have what is called a Gaussian or bell-shaped curve, where the large majority of the mice have this kind of average, kind of in-between color. Natural selection can drive populations to change in several ways. In the first way, it's called stabilizing selection. Excuse me. So natural selection selects for, I'm going to call it the average phenotype, right? That is called stabilizing. So it basically gets rid of anything in the extremes. A good example of that is human birth weight. So human birth rate, being very small is very, very bad for the baby. Being very, very big is also really bad for the mother. And very, very often then, at least historically, the mother might have died as well. So this has been stabilized towards the center. The second way that natural selection can drive populations is it can drive populations either in one direction or in the other direction. And that's called directional selection. So here, natural selection is selecting one extreme over the other. So it's going away from the stable middle and it's shifting to one extreme or the other. An example of that is in peppered moths. So in the Industrial Revolution, tree trunks went from nice and clean to being completely kind of covered in soot. And so what people saw is that moths that were darker colored survived better. All right. So they were kind of favored to be those moths that could hide better in these sooty trees. All right. And the third form, the way that natural selection can drive populations, is called disruptive. And that's when natural selection selects for both extremes over kind of the average, right? So here you're selecting for mice that are both light colored coats and dark colored coats. And examples of that have been frequently seen in birds, where birds with really tiny beaks survive and birds with big fat beaks survive due to food sources, but the middle beaks are selected against. All right, so Just as a reminder, we had mutation that can drive microevolution or changes in allele frequency, and we have natural selection. Our third here is migration, gene flow. This is just exactly as it sounds, the transfer of alleles / organisms from one population to another. Basically, the movement of fertile individuals or their gametes, right? So sometimes plants and stuff release their pollen or sperm into their environment, and that can move. So it doesn't have to be full organisms. So here, again, we have our little beetles, which are all homozygous recessive. And we have over here a population of beetles that are all homozygous dominant. And we are seeing migration of one beetle into the other. And now, if they're interbreeding, we will see a change in the allele frequencies, right? So migration or gene flow can drive changes of allele frequency over time, which is the definition of microevolution. The fourth thing that can drive changes in allele frequency are non-random mating. In other words, selective mating. And as you know, humans are very selective in their mating, right? As you know, you teenagers, you're not going to mate with anybody, right? You're very choosy, hopefully. So this means you show some sort of preference for mating for individuals within a population, all right? And we see this very often in many populations. So here are just two examples. One is assortative mating, preferences for organisms that look similar to each other. You can also have disortative mating, where you have a preference for different genotypes, right? One example that often comes up when we're talking about non-random mating are, I can't draw this very well, but you'll maybe see what I'm drawing. You see, this is like a really showy, fantastical bird. This is the peacock, right? So the peacock has this crazy plumage in order to attract mates, because the mates really like that fancy tail, right? So very often this leads to sexual dimorphism, where the males and the females of a species look different. And that is very often driven by selective mating. So selective mating can cause changes in allele frequencies over time, driving microevolution. And the last one is called genetic drift. This is a change in a population's allele frequency due to chance events, all right? And this is very drastic in small populations, right? So let's look at our little beetles. Here we have our little population of beetles with our allele frequencies. Due to a chance event, only 3 beetles, these ones circled, can have babies. The rest of them, let's say, have been swept away by a windstorm. And so that has completely changed our allele frequencies very dramatically, because this is a small population. So genetic drift, chance events, can cause changes in allele frequencies, driving microevolution. And I want to give 2 examples of that in the next slide. So 2 examples of genetic drift. The first is called a bottleneck. The second is called founder's effect. And again, genetic drift are chance events, changing allele frequency, and this is very dramatic in small populations. So the first here is bottleneck effect. So the best example of this is in our cheetah population. So hold on, let me get my note here so I don't screw this up. So cheetahs have less than 1% diversity, right? So about 10,000 years ago, there was a mass extinction event around the end of the last ice age, and only a few cheetahs, a handful of cheetahs, were able to survive. And so that handful of cheetahs, that very small amount of cheetahs, has then propagated to make the entire population. So really the definition is reduced genetic diversity after a severe reduction in population size. Another example of this are in elephant seals, which were nearly hunted to extinction. And therefore, they have a very small genetic variability in their population. So if you have a huge mix of things and a bottleneck only allows a few out, a few survivors, that then limits our genetic diversity. And often those populations cannot adapt as well because they have less going on. The second example is a founder's effect. This is where we have reduced genetic diversity when a small subset of a population goes and establishes kind of a new colony. So if we have a mother population, only a few of those move to a new environment and then will create an entire new population. And a great example of that is in the Amish, in a very particular place in Pennsylvania, where there was originally only 30 Swiss founders, and they mated only with one another. And one of them had this very interesting Ellis van Creveld syndrome, and which causes polydactyly. So a lot of this Amish population, actually 1 in 14, has extra fingers or extra toes, whereas that disease in that genetic disorder in the general population is about 1 in 1,000, right? So again, the 5 drivers of microevolution, evolution or changes in allele frequency are MN, MNG. Mutation, natural selection, migration. What's the other N? Help me, help me. Natural selection, migration, oh, non-random mating, and genetic drift, okay? And so Hardy-Weinberg, this is where we're gonna bring in Hardy-Weinberg. So Hardy-Weinberg are 2 mathematicians very interested in studying allele frequencies. And what they came up with was this equilibrium. It was a principle, a mathematical principle that states the genetic variation in a population will remain constant in the absence of any disturbing factors, right? And they have 5 basic assumptions to make this theorem work. And what you'll notice are most of them are the things that we just talked about that can drive evolution, right? So if these assumptions are met for a gene, then no allele frequency change will occur. These assumptions are very, very large population size, completely random mating, absolutely no mutation, no migration, and no natural selection occurring. And what you'll see, this is unlikely, right? This is very unlikely. But what this theorem allows us to do is look and see if allele frequencies are changing. And if they are, it allows us to try to figure out which of these factors is responsible for that. So the Hardy-Weinberg equilibrium or equation predicts genotype frequencies if the population is in equilibrium, if it is in Hardy-Weinberg equilibrium. So let me repeat that. Predicts genotype frequency if no changes in allele frequencies. All right. And by allele frequencies, I mean our P's and our Q's, which are always going to add up to equal 1. So let's look at this. We have a small population of beetles here. And let's go ahead and calculate their allele frequencies. So I'm going to always start with Q. Sorry, I don't like that up here. I'm going to start with Q, which is going to be our recessive allele little a. And if I calculate this, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 10 times 2. And we have, let's look at the hets. We have 1, 2, 3, 4, 5, 6, 7, 8. We have 8. Sorry. We have 8 heterozygotes. Sorry. So this is homozygous recessive. This is our heterozygotes. And we then have, that's going to be 8. So we're going to add those up. So we're going to have 28 over 40, which is our 0.7. is going to equal Q. And we can do the same for P. Again, calculating homozygous dominant, or the dominant allele rather. So if we look at our big A's, we're going to have 2 individuals here. So we're going to have 2 times 2. And for our hets, we already decided that we have 8 of those. So 8 times 1. And so that is going to give us 12 over 40 to give us 0.3 is our P. All right. All right. What you'll see now, if this population is not changing at all, we're going to see this equation where P is the frequency of the dominant allele, Q is the frequency of the little allele or the recessive allele, and this is the Hardy-Weinberg equation. P squared plus 2PQ plus Q squared equals 1. And what's really important here is that P is equal to frequency of the dominant allele. Q is the frequency of the recessive allele. And this now allows us to predict 2PQ and Q squared, this, but where P squared is equal to the frequency of homozygous dominant, 2PQ is going to be the frequency of the heterozygote, and Q squared is the frequency of homozygous recessive. All right, so this equation allows us to go from allele frequencies to genotype frequencies. All right. And so what we can do is if we have our P and Q, we can just plug and chug. We can do P squared and we can predict what our genotype frequency is. For a het, we're going to do 2 times P times Q, and we're going to predict what our heterozygous genotype frequency is. And we can do the same for the recessive, homozygous recessive genotype. So how did they come up with this? Something you guys already know about. So if we have, so here's our starting genotype frequencies that we just determined before, yes, and we have our starting allele frequencies. Remember, these alleles are going to go into this big old pool of gametes. And gametes combined at random. And remember, we know this by using our Punnett square. So when we do a Punnett square of 2 heterozygotes, which we've done a lot of, what you can see is that we get, this would be our P squared, 2 of those, one of those. We see that we have 2 of our heterozygotes, and we have 1 of our homozygous recessive. So this represents the genotype frequency, P squared. These represent our genotype 2PQ, and this one represents our Q squared. So Punnett squares help to determine what this Hardy-Weinberg equilibrium really is. All right. And again, allele and genotype frequencies stay the same over generations if those 5 assumptions are met, right? And those 5 assumptions that are met would be 1, infinite size, infinite population size. 2, completely random mating. 3, no mutation. 4, no migration. 5, no natural selection, right? So if none of those are happening, then our allele frequencies will stay the same. Okay, so the Hardy-Weinberg equilibrium allows us a way to measure if populations are evolving or not by comparing allele frequencies over time. Again, we have to know what our frequency of our dominant allele is and our frequency of our recessive allele. Then this allows us to predict genotype type frequencies. And that is using our p squared for the homozygous dominant, 2pq for the heterozygous, and q squared for a homozygous recessive. In this equation, p squared plus 2pq plus q squared equals 1. So this is the big equation that we're going to be using. And we will go over lots of practice questions with this, with how to use this in the future. Lots of little questions on the end here as well. All right. Peace out.